Hi everyone, I'm Allie Keene with the New York Stem Cell Foundation Research Institute. Welcome to our virtual classroom. We are so excited to have you with us. The New York Stem Cell Foundation Research Institute, or NYSCF as we like to call it, is located in New York City. At NYSCF, we study human stem cells in many ways to help us better understand and treat different diseases like Alzheimer's, multiple sclerosis, cancer, diabetes, and many more. Our amazing researchers are always hard at work. We have two of those researchers joining us here today, Dr. Valentina Fossati and Dr. Davide Marotta. We also have Jana Sotomayor joining us from Space Tango. Space Tango helped with the experiment you're going to learn all about today. They design, build, and operate automated facilities for research and manufacturing on orbit, so all things space. She's an expert. Without Space Tango, our cells wouldn't have made it all the way up to space, and she's going to tell you all about that. Along with our presenters, I have a few guests that are joining us. You'll see them answering questions in the Q&A. David and Reka are in the back end, so if you see their name pop up, they're just helping our presenters answer some of your questions. Before the presentation starts, I wanted to give a few quick reminders. Right now, you can hear me, but I can't hear you. It will be like this for the entire presentation. If you have any problems with the webinar, you can't hear us, you can't see us, please let us know in the participant chat and we'll make sure we get that fixed as fast as possible. We love your questions. There are two ways you can ask questions. At the bottom of your screen, you'll see a Q&A button. You can type your questions in there. You can also raise your hand using the little hand icon that you see at the bottom of your screen. This will let you know that you want to ask your question out loud. Today's presentation will be about 20 minutes long. Questions will be answered all throughout the presentation and at the end. NYSCF's virtual classroom is being recorded, so you'll be able to share it later. We also have a number of other virtual events coming up that we welcome you to join us for, so check back to nyscf.org often as we continue to add them. Now I'm excited to hand things over to Dr. Davide Marotta.
Hi, hello everyone. Thank you for the amazing presentation. Hello everyone, and welcome to today's session. I'd like to introduce today's presenters, two amazing women in science, Valentina, brain scientist from the New York Stem Cell Foundation, and Jenna, a space scientist from Space Tango. This is Davide, and I'm also a scientist, a brain scientist working with Valentina and NYSCF. For those of you just joining us, welcome, and let's start this amazing journey to space. We all know that astronauts travel to the space station to work, live, and explore life in space. But what is the International Space Station? The International Space Station is the largest spacecraft orbiting Earth, home for astronauts in a science lab. NASA is learning more about living and working in space, and these lessons will make it possible to send humans farther into space than ever before. The space station was born from the idea that humans can live in space where conditions are difficult and extreme. More than 15 countries altogether planned and built the space station. Its assembly started in 1998, and the first humans arrived 2 years later, and only in 2010 it was completed. It took 10 years and more than 30 missions to assemble. Quite incredible, right? The space station is a huge spacecraft, but do you know how big is it? Well, the space station is as big as a football field and weighs 1 million pounds, which is 358 times heavier than a car. The big station flying around Earth is extremely important to study the human body in space, make scientific discoveries that will help life on Earth, helping humans to reach faraway worlds like Mars. This big station flying around Earth It's very fast. It can fly Earth in just 1 hour and a half. And per day, people on board of the space station can see 16 sunrises and 16 sunsets. Now, imagine looking out of your window and seeing the sun going up and down 16 times in a day. It might be really hard for you to sleep, right? Well, this is what the astronauts are experiencing on board of the space station. And if you're curious to know when the space station will fly above you, you can check this link. So the station travels 17,500 miles per hour, which is equivalent to 28,000 kilometers per hour. It's basically 30 times faster than a plane and 350 times faster than a car and 6,000 times faster than a human. Oh, yes, Sonic, no, faster than you. On Earth, we can walk, jump, play, and do many other activities, but because we are on the ground. But what glues you to the ground? It's gravity. Gravity glues you to the ground. Gravity is that force that pulls you down when you jump high in the sky from your trampoline. But in space is a different story. Gravity doesn't have much power, and life there is quite different. For example, washing your hair could be a quite complicated task, and walking, well, you don't really walk in space, you float. But now let's explore with Jana how different is life on board of the International Space Station. And now, Jana, it's time for your liftoff.
Whoop, Jan, I think you're muted.
Thank you, Dr. Marotta. It's very great to have the opportunity to speak with all of you today. And I'm very pleased to talk to you not only about what happens to the human body in space, but how we get to the International Space Station with experiments. Next slide. Thanks. So when you go to space, not only do you see different night and day cycles, but you have to think about sleeping in space is not like sleeping in your own bed because you're floating. So you would have to be Velcroed to the wall. And you can see the astronauts have special sleeping bags to help them sleep up there. They also don't have any of the fluid that would normally go down into their legs falling down. Instead, it goes up into their heads. And it actually can make their faces change. When you look at an astronaut's picture on the ground and then you see an astronaut in space, you see that their face is puffy because the fluid has shifted up to their head. It affects their eyesight sometimes too. And because they're floating, they don't really use their muscles and bones quite as much. So they have to exercise about 4 hours every day just to stay in the same shape that they were in when they launched. So what happens in microgravity and why is it so different? Well, when you were thinking about washing your hair, think about washing your face and not having the water drip off into the sink. What if it just stayed right on your face? That's what happens in microgravity. Surface tension is different. The way that fluids actually stick to surfaces is different. If you boiled something in space, the bubbles wouldn't go to the top, they would stay right on the bottom because there's no forces to actually lift them back up. And your birthday candles that are on your cake, they would have little round blue balls instead of nice long yellow flames because no heat rises when you're in microgravity. The same thing happens to cells. The way that cells signal to one another, the shape that they take on, the way that their genes might change in space. That's all different because they don't have that force acting on them that would be acting here on the ground. So it's a perfect opportunity for us to be able to study systems in that environment where we can take out a variable that we could never take out here on the ground. I'm going to show you a little bit now about where we work. So to get to our offices, we take a rocket and we go up to the International Space Station with our payloads packed in the Dragon capsule. Once they're unpacked, they're installed into facilities that are on board the International Space Station that we can control from the ground. And what's really kind of cool about rocket launches, you know, you probably always hear the 10, 9, 8 countdown, but one of the most important things in a space liftoff is waiting for the main and secondary engine cutoffs. Once those rockets and capsules separate, you've all seen the SpaceX Falcon 9 rocket come back down and land on land, which is fantastic to see. But that means that our Dragon capsule is well on its way to the space station and bringing with it supplies for the astronauts, food, sanitary supplies like new toothpaste that they might want, sometimes special treats like ice cream, along with our research. So we build little houses for the brain organoids that we sent to the International Space Station. They're automated so that we don't have to have astronaut hands changing the media, trying to do the experiments on station for us. And when you think about it, when we were talking about how fluids move differently, you can't really just open the lid of a petri dish in space. All of that liquid would just come right out. So we keep them in closed systems that we use pumps and heaters to keep them warm and fed and in a perfect environment to be able to be cultured on station. Once they're in our facilities on orbit, we then can control from the ground what happens. We can see those cells, we can decide we might need to feed them a little bit sooner or a little bit later, but we have complete control of the experiment, and it eventually is repackaged back into that Dragon capsule and brought back down so that the teams can then do more analysis once it returns. Here's an example of just a few of the things that we can do on the space station. You can see plants that are growing on the left-hand side of your screen. And to the right, you're going to start to see an experiment that we did on separating two different types of fluids on station. So remember when we talked about how fluids act very differently? Trying to separate them in space is sometimes very hard. But that's something that's really interesting to look at in microgravity. And up at the top, we've actually even flown bees. And I'm going to tell you a secret. When bees get up there, they learn very quickly that they don't actually have to flap their wings to fly. Here's an example of some of the science payloads that we work on for tissue chips. Tissue chips are human organ systems, but they're more like a carpet of cells. They're not quite the same as the organoids that we studied in the project that we did with the team at JAX-NYSCF. But they're a great example of a different way for us to be able to look at human body systems in microgravity. And here's the actual flight hardware that the brain organoids flew up to the International Space Station on. As you can see, it's a pretty complicated system once it's all together, and there were lots of brain organoids that were put in there. And the picture at the bottom, which Dr. Fossati will tell you a little bit more about, starts to show you exactly what types of cells we flew in those brain organoids and why the research in microgravity was important.
Yes.
Dr. Fossati?
I'm taking from it. Thank you, Jenna. So you see here an actual video of some cells that we have colored with the red and green to be able to recognize them. But what exactly are these cells? So as a scientist, I work on the brain. I'm trying to understand with my colleagues how the brain functions, because we want to be able to fix when there are mistakes in many diseases, and we want to be able to cure these diseases. So to do that, we need to study the human brain, but we can't really go around and ask people, hey, can I get your head? Nobody would give me their head, right? So the way we came up is, a few years ago, some scientists found a way to actually make the same cells that are in the brain, but make them in a dish. And we make them in something that we call the organoids, which are basically a much simpler version of the brain, or any other organ, actually. That could be the kidney, the heart. And with these tiny cells, we can look inside. It's really tiny. You can see here how one real organoid looks like from the microscope. We usually look at the microscope, but you can also see them. They're visible. This is my hand holding a tube. And if you look carefully, you can count some tiny white dots. Each dot contains millions of cells. Cells are really, really small. They're so small, but yet they're really important because they really tell us many, many informations about how the brain functions. And inside the brain, there are several different cell types. I think most of you are familiar with the cells that we call neurons, or also nerve cells. Neurons are basically what allow us to do everything we do. They allow us to move our fingers, to think, to talk, because they are signaling all the other part of the bodies. They are sending electric signals for what to do. But they're not alone in the brain. There are many other cell types, which are called glia. And one of these is called microglia. Micro, it's from the Greek and means small. So those are tiny cells, very important. They have a lot of fingers, and they keep moving these fingers all the time, scanning what's going on. We could say that they are the police officers in our brain, because they keep looking around and trying to find the bad guys. The bad guy can be, for example, a virus. And I think all of you now are very familiar with viruses. Sometimes they enter our body, some of them like the lungs. Some of them enter in our brain and they make us sick, and we don't want that. So microglia are there to be able to find a virus or find a bacteria or find a cell that is dead and get rid of it. And they literally eat it. They eat it, and though they clear the environment. And this way the neurons can keep being healthy and can keep doing their job. So those are really important cells. We can see here a real video of cells that we grew in a dish we were able to make, and just focus on one. Each of one is one cell. You see how they move? They're like little crabs going around, and you see how they really change the shape, and sometimes they touch each other, and we can replay the video. So look at one, how they are really, really elongated, and then they become smaller, but they keep moving around. They are always searching for the bad guy and making sure that everything looks fine inside the brain. This is another picture that we took of a real, this time is an organoid. So here we have in red all the neurons, and in green we have the microglia cells. This is, we use a very sophisticated software that we have in our microscope. So we are able to transform a picture and make it becoming a three-dimensional image. So you can really see the shape of one of each of the microglia cells. They're really cool, right? And so we took these organoids where we had neurons and microglia together, and with Davide and Jenna and everybody in the team, we said, why don't we have them going for a trip? Why don't we send them to space and see what happens when they are there? And Jenna just told you how many things are really changing when you are in space and there is no gravity. So what's going to happen to these cells? Well, to do the experiment, it was actually a big trip, a major trip, even on Earth, because our laboratory is based in New York City. But the SpaceX rocket launches from Florida. So the cells had to reach Florida first. And to do that, we ship them the same way as you would ship any gift, or you are doing online shopping. And actually it was more complicated than we expected, because the first time we did this launch in July, the cells got stuck in Texas. But luckily, everything worked well, because we don't ship them with regular boxes. We take some very sophisticated boxes that are keeping the cells cool. They're basically a mini refrigerator. And so everything was able to arrive and was loaded inside the rocket and was launched in space. And I told you the first time we did the launch was the SpaceX 18 mission, which was in July. And that was a test experiment. Why do we need to do a test first? Well, Jaina told you that Space Tango built those little houses for the cells, and they literally built it just for us. Each experiment has a unique house. So it was very important to be able to first see that the house would work well before we actually can collect data on our experiments, on how our cells behave. So here in the first picture, you see us all prepared in the lab with other scientists. We finally have the house ready. And then you see a picture with me. This is my hand holding a microphone. This is because NASA, on the day of the mission, organized What's on Board, inviting the scientists that are doing some research that are shipping experiments so that everybody can know what's gonna go up. And there is, you can watch on the website that the NASA is still there. So if you're interested and you wanna know more about other experiments that went up, you can certainly follow that. I think kids were very excited in July because one of the experiments that went up with our cells was the slime. I'm sure that astronauts had a lot of fun playing with the slime, although we don't know yet what happened to it in absence of gravity. This is also us again finishing up, preparing our cells, and then the cells got into this exact rocket in July. And then once everything was tested and worked well, we got ready for the second experiment, which was under the SpaceX 19 mission. It was the past December. And here we are assembling again our house, and we brought our names. So we didn't go up to space, but our names did, and we were very proud. And this is the actual launch as it occurs, and we were lucky to see it directly from there. You can listen to the video. So this is SpaceX 19 going up. And as Jenna was saying, the capsule reaches the ISS, where it gets captured, literally catched by a robotic arm. And then the astronauts are able to access the cargo. It's a huge cargo. More than 4,000 pounds of supplies and experiments were up. Imagine the astronauts having, it's like you're going shopping and you have hundreds and hundreds of bags. So they have to take everything out. And in our case, they had our little house and they had to put the power on it. And then it was fully automated because it's like working like a robot. You can see here probably when it just came out of the cargo and was floating around. And then they kept it there together with many other experiments for 30 days, around 1 month at the ISS. After 30 days, it has to come back down because we want to now see the results. So the astronauts are, unplugging it and preparing it for shipping. And I don't know if you recognize, if you know the astronauts that are up in the ISS, but David and I were extremely happy and proud that Luca Parmitano, it's an Italian astronaut, was there since we are both Italians. And so we were honored that he would, take care of our experiment as well. So Luca and his colleagues re-prepared everything for shipping it back. And everything was loaded inside the capsule, and the capsule Dragon was splashed down in the Pacific Ocean. It's about 400 miles from the coast, where there is the SpaceX team that is waiting, and then they hoisted it into a recovery ship, and they brought it back to the port of Los Angeles. This picture that you see is actually the last one ever, because this SpaceX capsule is retiring and is gonna leave the space to a new capsule that is gonna be more advanced than this one. And now that we finally got back our organoids inside their tiny containers, Davide has started all the analysis. Basically, we're gonna do what you saw before. We're gonna take picture of these cells and we're gonna try to look, how the cells are now. And we are gonna also compare them with the same experiment that we did, but here on Earth, because obviously scientists have to work, with a very precise method. So we run the same exact experiment with the same exact house, but on Earth instead of the ISS. We are showing you here the very first picture that Davide took. We don't have yet any results because this is gonna take a long time, but we are starting taking images of how the cells, how the organoids looked in space and how the organoid looks in the ground control. And for the one of you that are more familiar with science, they have studied already in school some biology, we will also look inside each of the cell at the DNA, of the RNA, at the gene expression, so that we'll be really able to understand what happened during this 1 month without gravity. And we are very excited about this experiment, and we are already thinking of what's next. And we are very much looking forward to continuing our missions. This is to show you how many people were involved in this mission. It really takes a village. Many of us contributed, but also is to show you that we were people from very different backgrounds. I'm a biologist, there were engineers, chemists, and so if you really love space, you don't necessarily need to be an astronaut to be able to involve with science in space. So we hope that you really were excited about this class, and maybe in the future we will see some of your experiments. Thank you all.
Thank you, Valentina, Jana, and Davide so much. So we're gonna do a quick Q&A. Well, we have some time for a Q&A actually. I'm gonna do one quick question first. Davide, you might need to pull up your presentation really quick for it, but if not, what does the red and green mean in the picture of the cells? What were those explaining?
So those were the two different cell types. I think you're referring to the first video where we have the microglia cells with the neurons. So if you're able, Davide, to replay the video, the neurons are there and they have this very long, axons and dendrites. So they're really creating a network and they don't move, they stay like that. But the microglia, as you can see, they move, they really love moving. So the video shows you when we attach the microglia to the neurons, the red cells are going deep inside and they're crawling and they're basically moving all around. And this is, what we want to see basically if it changes when we go in space, because they're gonna be able to move in a different way. The same, like astronauts are not able to walk, but they're just floating around, how the cells are gonna move without gravity.
Cool, thank you. So you mentioned how long they're at the space station. How long does it take for the cells to get to the International Space Station once they leave the ground?
Yeah, Jana, do you want to say more details about that?
Sure. And so when you load a rocket, you have to think about it's just like a big moving van, right? When Valentina was talking about how much cargo goes up in each one of those missions. So there's different times that they start loading the vehicle, but for our payloads, we load about 24 hours before launch because we have to turn our payload over, it gets put into the Dragon capsule, and then the capsule gets attached to the rocket is stood up, it's fueled, and then it's launched. And once it's launched, it takes about 2 to 3 days to get up to the space station. And once it's at the space station and docked, it takes a few hours for the pressure to equilibrate after it's been docked to the station before they can actually open that hatch. So we try within 72 hours of launch to be able to have all of experiments in our facilities on orbit so that we don't have any delay in getting those cells into a place where we can actually see them. And you're probably wondering on splashdown, that takes about 6 hours to drop down from the International Space Station into the ocean. And it takes a little bit longer than that, usually almost a day to tow into the dock, but you're in the water 6 hours after you're released from the ISS.
Cool. We have a question from one of our students, Sarara. Have any other cells other than human cells been sent up to the ISS?
So lots of different types of cells have been sent to ISS. And in fact, we send all kinds of organisms. So flatworms have gone up. Rodents actually fly on the International Space Station. It's not something that Space Tango actually supports, but rodents fly all the time. All kinds of different yeast and bacteria. There has been some work that's been done with frogs and fish before as well. So almost anything that you can imagine, except for birds and pigs. Pigs don't fly quite yet, but at some point we may be able to have larger animals as we have more space as well.
Cool. This is maybe for Valentina or Davide. Did the cells grow faster in space?
Yeah, we don't know yet, but there were some experiments done before with other cell types, not exactly with the brain organoids, that showed that cells did grow more, but we will see what happens with our cells. Yes, that was a very good question. It's very important to know that.
Yeah. How many cells did you send up to the ISS? Was it 1? Was it 1,000,000?
David, do you want to answer? It's a lot.
Oh, Davide, I think you're muted still. There we go.
Yeah, there were a lot of cells. I think we plated 1 million microglia per organoid, something like that. There were a lot, and we had like about 64 organoids on the space station and 64 on Earth. So there were a lot of cells.
That's a lot of cells, but they don't take up much space, right?
No, they're pretty tiny.
All right, I'm going to allow one of our attendees to ask a question out loud. We have Liz who's going to ask her question. Liz, I'm going to allow you to talk. Can you hear us?
Yes.
There you go.
I have a question about why you use stem cells. Why did we send these organoids that were made from stem cells to space versus all the other things that Jana was talking about, like rodents or other animals? Why send stem cells?
Yeah, so, you know, that's a really important point because those are human cells. And Dr. Fossati and Dr. Morota can actually talk more to you about what induced pluripotent stem cells or stem cells that are taken from your body. Why they're so important is we don't have good human models. And even when we test in animals, it's helpful, but it doesn't necessarily tell us exactly what it's going to look like in a human. So stem cells are a really important advance in helping us to understand exactly what's happening in human tissue.
Yes, if I can continue, we are very interested, as I said, in studying the brain, and our group is interested in understanding diseases such as Alzheimer's disease or Parkinson's disease, multiple sclerosis, and the role of microglia in these diseases and why they stop doing basically their job during when the brain gets sick. And so the only way to be able to study these diseases is to look at human cells. And so we really use this powerful new system that was discovered less than 10 years ago. So when I studied, none of this was available. This was complete science fiction. It was impossible to make human cells in a dish. But 10 years ago, this became possible. And so now we are really exploiting this new system to understand human diseases.
Great, thank you. We're going to allow one more question, a couple more questions from our attendees. So Anna, I know, has had her hand up from the very beginning. Thank you so much for being patient. Can you hear us, Anna?
Yeah. So I have a question. How did the cells look like when it was after it was from space?
So what did the cells look like after they came back down?
Can I answer?
Yes.
Yeah. Hi, Anna. Thank you for your questions. With a naked eye, I was not able to see any difference, you know, because they are so tiny that I should have a superpower to look into them. So I used the microscope. And one thing I can tell you that there were some differences in the size of the brain organoids. So, and now I'm going to look into the organoids deeper using the microscope and the imaging system you saw in the slides, and soon we will have the answer.
So hopefully we'll be able to answer that question soon, and we'll keep you all posted. Rohan, do you have a question for us? Can you hear us? Oh, Rohan, do you have a question for us out loud?
Yeah.
What's your question?
So you said that astronauts have to expect the equipment to they use.
Oh, I can't really hear you. Can you try one more time?
So what type of equipment do they use in space? Because they have to exercise for 4 hours a day.
So the picture that I showed you, did you see how they had a belt on that actually strapped them down to a treadmill? So they have a treadmill, which allows them to run in space. They also have a weight machine that's pretty cool, because when you think about how everything is weightless in space, you can't really lift weights, right? Unless there's some resistance force to it. So their weight machines actually look a little different. They look kind of the same as what you would see on the ground, but instead of thinking about the way you would push up a weight, it's more of a pull so that they can get a resistance exercise while they're up there. And we have had an astronaut who actually ran a whole marathon on the treadmill up there once.
Oh, that's like people running marathons in their backyards right now. We have a question from Luke. Have any of you ever actually been in a space rocket?
No.
Now,
I'm lucky enough to have been able to sit in simulators and in vehicles while they've been being built, but nothing that's been on top yet.
Yeah, we all did the simulation. If you go to KSC, you can do it. It's 5 minutes. I have to say, it was enough for me. I'm not particularly, but it's exciting. But it needs a lot of training. Astronauts undergo a really strong training to go up. So it's not that you can just decide, hey, I want to go tomorrow. It really years of training, right, Jenna?
Absolutely. But when you think about what's going to be happening, May 27th, we're going to actually launch US astronauts from Kennedy Space Center again for the first time in a long time. So we're all very excited about that opportunity. And there are going to be commercial crew opportunities, which means someday as a scientist, you may actually be able to fly alongside your experiment as well. And that's the not so distant.
By the time and some of you become scientists, I think that will be a possibility.
Absolutely.
yeah, definitely.
Well, hopefully one day we'll see you guys up in space. What's the most harmful effect of microgravity in cells based on the research you've done so far?
So I wouldn't say it's the microgravity, but there is a radiation effect on station as well. When you think about it, you're 250 miles up, so you see a little bit different type of radiation than you would see here on the ground. And when you think about it, it's about the same as what an airline pilot might experience by flying at 30,000 feet. You just happen to be 250 miles up. So it's a little bit more intense, but that radiation effect, and it's one of the reasons why it's so important to have these types of human models that we can study, because as we want to go further, we need those models to tell us how to develop countermeasures for things like stronger radiation that we're going to experience when we go to the moon and Mars. And that's not so far away either. I mean, we're landing the first woman and next man on the moon in 2024. So that's coming.
That's not too far away.
Nope.
We have a question from Candace, who is 5 years old, and she said, why is there no gravity in space?
Oh, so that's a really good question. So when you think about the International Space Station, and remember how Davide said that it's orbiting around the Earth, right? And it's going really, really fast. So when you're in that orbit, you actually are experiencing an effect where all of that force of gravity that would be holding you down is gone. And in microgravity, space is pretty much a vacuum. So you really don't need to have anything that keeps you in one place. And if you watch when the astronauts do their spacewalk, you can see when they hold on to the rails outside, if they let go, they just kind of float right in the spot where they are. And it's actually pretty fun to hear them talk about how they train in a big pool in Houston. If you ever get a chance to go to Johnson Space Center, they have a replica of the ISS segment in this massive pool, and they put them in their spacesuits and they put them in the pool. And they talk about how being in a pool and floating, it's a sense of weightlessness, but it's not quite the same as being in space. When you really step outside into space, you can hold on by one finger. And they often don't figure that out until they've gripped very hard for the first hour of their spacewalk, and they realize they can just let go.
I'm going to do a couple more questions. I know we're running a little short on time, but we have so many students asking questions, so we're going to ask a few more. So Maria has a question. Do we know yet from the cells how longevity is affected in space or microgravity?
Not yet, but this is a very important question and everybody wants to answer that. So maybe in one of our next missions.
But the twin study, which was really interesting, it was a 1-year study with 2 twins, one that was on station for a year and one that was on the ground, tried to look at some of those effects and to see what those differences. Station is in many ways, a model for accelerated aging. Just like we talked about, you lose bone mass and you lose muscle. Your muscles are just not quite as robust because they're not used. So some of that we are learning, but we have a lot more to go.
Great. Let me just review these questions really quick. This is a question from Steve. Do you have any plans to send more brain organoids to the International Space Station?
Yes, we do. So hopefully we are continuing soon.
So as you saw, they still have a lot of questions that want to be answered, just like you guys are asking. They're asking the same questions. So hopefully they can send some more. So I know we're running a little bit out of time. You guys also, all the students have such great questions, but I'll send a link around so that you can continue to ask those questions and we'll get them answered from some of our scientists. So I have one question left. I know that we didn't do this alone, and there's a lot of people involved in this research. How is the work supported, and are there other groups involved that are helping with this experiment?
Yeah, Jenna, do you want to say? Yeah.
Sure. Well, so this project was really the vision of the National Stem Cell Foundation. And Dr. Paula Grisanti has been phenomenal in her vision of thinking about how do I take the best scientists that I have and how do I use new tools like science in microgravity to be able to answer really hard questions on diseases that we can't really solve, like Parkinson's and MS. And so when she came to sort of Space Tango and talked about the fact that she had brilliant research teams at NYSCF and teams in California that she wanted to give this opportunity to, we were super excited because the opportunity for us to work with leading experts in the field and give them that opportunity to use that platform for future discovery is just fantastic.
Thank you. Well, we're so excited that we're part of this, and I was there when we launched some of them and it was very cool. So be sure to check out online when those go up. They're really cool to listen to. You can hear NASA calling out the countdown. So I just wanted to say thank you to Valentina, Davide, and Jana for teaching us all about space and the experiments you are working on. As I mentioned, if you have more questions for NYSCF scientists, I'll be sending a follow-up email and it will include a link where you can ask all of your questions and we'll try to answer those as best we can to all of you. We hope you had a great time this afternoon. Be sure to check back to NYSCF.org for more virtual classrooms from some of our awesome scientists. There will be a brief survey right after this, and we'd love to hear from you so we know what classes you want to learn more about. Don't forget to tell your friends and families all about what you learned today, and we hope to see you soon. We also want to say a quick thank you to the Stavros Niarchos Foundation for making these classes possible for students. We're so thankful, and we hope to see you soon. Bye, everybody.
Bye.
Bye.
Bye.